To keep a single human cell alive, let alone rapidly expanding and dividing like this, requires an immense, continuous expenditure of energy. We often reduce the word metabolism to how quickly our bodies burn calories. But strictly speaking, metabolism is the complete, total network of life-sustaining chemical reactions occurring inside a cell. That network is split into two opposing halves. The first is catabolism. This is an oxidative, degradative process. It breaks down large molecules into simpler products, releasing trapped energy. You observe this directly during digestion. A large fat molecule is broken apart, severing its chemical bonds to leave distinct fatty acids and glycerol. The second, opposing half of the network is anabolism. This is a reductive, constructive process. It consumes energy to fuse small, independent molecules together to build cellular materials. This is how individual amino acids are assembled into proteins for tissue repair, or how raw nucleotides are chained together to construct a DNA helix. Cellular survival relies completely on these two forces. Destruction and construction must operate in a continuous, perfectly balanced flow. This is a strict thermodynamic problem. How exactly does a cell capture energy released during destructive breakdown and transport it to fund anabolic synthesis? The solution is a physical and chemical bridge, a universal energy currency called adenosine triphosphate, or ATP. This diagram displays the physical structure of ATP. It consists of an adenine base, a ribose sugar, and crucially, a tail of 3 phosphate groups. When an external water molecule collides with ATP, it severs that third phosphate group in a hydrolysis reaction. Breaking this bond releases a massive burst of outward energy, leaving behind a depleted molecule known as ADP. We categorize chemical reactions by how they handle this energy transfer. Exergonic reactions, like catabolic breakdown, release energy. Endergonic reactions, like anabolic construction, consume it. The cell runs a continuous recycling loop. The energy released from exergonic catabolism on the left is captured to forcefully attach a third phosphate group back onto depleted ADP. This fully charged ATP then travels to the right, where endergonic anabolic reactions break it back down to fuel cellular growth. ATP serves as the physical link between these two systems. If this cycle of charging and depleting halts even for a moment, anabolic growth stops entirely and the cell dies. Extracting the energy needed to charge those ATP molecules relies on a highly conserved 3-stage architectural sequence designed to maximize yield. Stage 1 is digestion. Massive macromolecules, the carbohydrates, lipids, and proteins we consume, are broken down into their base monomers. In stage 2, those simple monomers enter the cell. We will trace the path of a single 6-carbon glucose molecule drifting into the cellular cytoplasm. Here, we see that 6-carbon glucose ring. It is entering the first major biochemical pathway, glycolysis. This process operates completely anaerobically, meaning it requires 0 oxygen to proceed. The violent mechanical action of glycolysis splits the 6-carbon glucose into 2 separate 3-carbon molecules known as pyruvate. The immediate net yield of this split is exactly 2 molecules of ATP, but it also initiates a critical process of electron extraction. The split strips high-energy electrons from the carbon chains, binding them to a coenzyme called NAD+ to form NADH. To finish stage 2, these pyruvate molecules are transported across a membrane into the mitochondria. During transport, each chain loses 1 carbon atom as carbon dioxide waste, resulting in a 2-carbon intermediate called acetyl-CoA. Glycolysis produces a minimal amount of ATP. Its true purpose is prepping the carbon skeleton and extracting the first high-energy electrons to feed the cell's main engine. That main engine is located inside the inner matrix of the mitochondria. This is the site of stage 3, final oxidation. Those 2-carbon acetyl-CoA molecules feed directly into a rotating pathway of chemical nodes known as the Krebs cycle. As the cycle turns, its primary function is to fully oxidize the remaining carbon from the original glucose molecule, ejecting it as carbon dioxide waste. The true value of the cycle is mass electron extraction. A rapid barrage of electrons is stripped from the central pathway and transferred to coenzymes, yielding a massive payload of NADH and FADH2. These loaded electron carriers immediately migrate forward toward the inner mitochondrial membrane to initiate the electron transport chain. In this chain, electrons hop down a sequence of specialized membrane proteins. The proteins use the energy from these passing electrons to pump protons across the boundary, clustering them densely on one side to create a severe pressure gradient. During oxidative phosphorylation, those trapped protons seek equilibrium. They violently rush back downward through a specialized turbine enzyme called ATP synthase. This mechanical turbine action rapidly forces unattached phosphate groups onto waiting ADP molecules. The final payoff is a yield of approximately 30 to 32 ATP per original glucose molecule. This multi-stage oxidative funnel systematically squeezes the absolute maximum potential energy out of a single sugar molecule. Now, the cell must deploy that newly minted ATP to power anabolic construction. One of the primary anabolic pathways is protein synthesis, which takes place on the cellular ribosomes. Linking simple amino acids together requires immense energy. A fully charged ATP molecule must be broken apart to weld every single peptide bond in the chain. Other crucial anabolic pathways operate exactly the same way. The liver expends ATP during glycogenesis to link glucose into storage chains, and adipose tissue uses it during lipogenesis to build fats. Because both halves of the system are metabolically expensive, the body requires strict regulation. It must know exactly when to run catabolic breakdown and when to switch to anabolic storage. This is managed through chemical switches. Insulin acts as an anabolic signal. When external resources are high, insulin triggers the cell to build and store. By contrast, hormones like glucagon and adrenaline act as catabolic signals. When energy is low, they force the cell to halt building operations and begin breaking down its stored reserves. Metabolism is a dynamic, highly responsive system entirely dictated by the chemical signals reflecting environmental availability. Under normal conditions, hormonal signals ensure that catabolism and anabolism perfectly balance the cellular economy. However, a severe external stressor, like starvation, forces the body strictly into a prolonged catabolic state in order to survive. The body executes a strict sequential breakdown of its reserves. First, it drains the Tier 1 glycogen stores in the liver. Once those are empty, it moves to Tier 2, draining fat reserves from adipose tissue to feed the ATP generator. When the fat is gone, the cell reaches the grim final stage of starvation. It begins catabolizing Tier 3, the body's own structural muscle proteins, sacrificing functional tissue just to keep the ATP engine running. We see a different kind of pathology when internal regulation fails entirely, as is the case with cancer cells. Cancer cells exhibit extremely high metabolic rates, but they alter the standard engine to support their rapid anabolic growth in a phenomenon known as the Warburg effect. On the left of this diagram, a normal cell routes glucose through the mitochondria for maximum yield. But on the right, the cancer cell frequently bypasses the highly efficient mitochondria entirely. It relies on rapid, inefficient glycolysis looping directly in the cytoplasm to secure its energy. Every biological action, from the slow repair of damaged tissue to the frantic growth of a tumor, is entirely beholden to the mechanics of this biochemical engine and the precise transfer of ATP.